Thank you. Thank you so much for coming, and I'm really delighted to be here. So I should tell you all that I studied in Moscow for 1 year when I was 19, a long time ago. So I do understand some Russian, but I cannot speak it any longer, unfortunately, but it was a great experience. And I should say that Anna and I have met throughout the years, actually, at various conferences, and we always try to find the collaborative projects, and we're getting closer and closer, actually. And we also had a lot of interesting discussions. So thank you for the opportunity to present our work. So I will give a general introduction to the field which we are studying, and then I will take two examples, two different diseases. And I should say straight off that I am, an experimental researcher. I don't see patients, so it will be from an experimental research angle. The study I show you. Right. So just to show you that this is Karolinska Institutet right now. It really changed the skyline of Stockholm. So we're building a lot of new buildings. It will probably be for the better. We are not absolutely sure yet, but we think so. And we are, among other things, we are building a new university hospital with all the things coming with that, and it's completely new for all the people that are working there. But I think, within 1 year, we're going to be up and running. And we're also building new research buildings. And to show you one of the buildings that are actually finished is our new lecture hall, the Aula, which is a donation from a private family in Sweden. You may have bought clothes at H&M, actually. So they have donated this to Karolinska Institutet. And what I like is that they also kept the oldest building at the campus in front of this modern building. We love that sort of thing. So my group is interested in immunodeficiency diseases, and we're trying to understand what goes wrong in these diseases. So you all know that the immune system needs to be at balance to be able to recognize any given pathogen we are exposed to, but also to remain innocuous to substances we should not react to, such as allergens and other things in our environment, what we eat, for example. When the immune system is at imbalance, we see development of common diseases, allergy, autoimmunity, and cancer. And these diseases are also common in patients that lack a certain part of the immune system, so the primary immunodeficiency diseases. And one of these diseases is viscodolic syndrome that both Anna and I have a long-term interest in studying. You know better than me, but these patients often come to the clinic with major recurrent infections. And for immunologists, these diseases raises a conundrum, and that is how can an immune system fail to respond to non-self pathogen while reacting vigorously to autoantigens and allergens and also develop cancer? So to answer this question, my lab has focused on immunodeficiencies caused by mutations in actin regulators. And why is that? Well, the immune system is never at rest. So our immune cells constantly survey the body, they go out to the tissue, they kill pathogens, or they kill tumor cells, and then they stop and go back to sort of status quo. And to do that, it's extremely important that they quickly can remodel their actin cytoskeleton when they go into the tissue and when they communicate with other cells through the immunological synapse. And the vast family of proteins regulate actin polymerization. And just to show you some examples, this is a dendritic cell moving in this direction, and for a cell to move, you need both to adhere to a substratum and generate force to move forward. So the dendritic cell have a leading edge lamellopodium, which is rich in actin. It also makes adhesion points towards the substratum called protosomes. All of these contain dynamic F-actin. And here you see an immune synapse, and the reason it's slightly blurry is because we have used image screen where we combine flow cytometry with imaging. And here you see an NK cell that will kill this tumor cell, and they make stable contacts via a ring of polymerized actin, and then they release the granules into the tumor cells to kill them. These granules contain a lot of cytotoxic enzymes. And it's also interesting that a lot of pathogens actually use the host cell cytoskeleton, especially the actin cytoskeleton, to really get into the cell, propagate through the cell, and infect the next cell. So here you see vaccinia virus in red that form these long actin tails. When they sort of move through the cell, they may infect the next cell and so forth. So actin nucleation is quite a slow process. So to make an actin monomer into a filament is very slow without any help from other proteins. So two main families in all eukaryotic cells can polymerize actin. Formings will create these long straight filaments. The WASP family of proteins will bind to the ARP2-3 complex, where ARP2 and ARP3 share a lot of homology to actin, and in this way they can make branched filaments, so very dynamic filaments. And the WASP family itself was, as probably many of you know, was originally identified, or I should say the WASP protein, by two clinicians, Wiskott and Nordisch, and this was in 1937 and 1954, that they described boys that were affected with a really severe immunodeficiency disease. In 1994, a team of researchers cloned the gene, and that was encoding Wiskott syndrome protein. And because of the homology in this region, the VCA, Vertolin Coughlin acidic domain, there were several more members in this family identified. So NWASP, that is very homologous to WASP, WAVE, and these are the yeast and the C. elegans homologs. But it's an evolutionary conserved family based on the VCA domain, and then the N-terminal differs somehow. And I should say that WASP is uniquely expressed in hematopoietic cells, so in immune cells. And they signal to the actin cytoskeleton via upstream CDC42, RAC1, RAC2, rhodidinases, and above them we have the guanine exchange factors, DOC8, Y1, 2, 3, and then they signal from different receptors on hematopoietic cells. And I should say that there are now, we have identified quite many immunodeficient patients that have mutations in DOC8. There are rare variants of RAC2 deficiency. WASP is sort of the focus of part of my talk. I don't know if there are others now, Anna. Is it other now? Do we add to the list?
Maybe WASP is embryonic.
Yeah, exactly. No. So maybe we see about 1, 2, 3. Yeah. Yes. So then WASP itself. So if this is for example, a T cell receptor, B cell receptor, WASP resides in an inactive form in the cytoplasm. Upon binding to CDC42 and also PIP2, it's recruited to membrane, where it opens up, exposes the VCA domain that induces actin polymerization. And this is needed now for immune cell migration, cell-to-cell communication, intracellular signaling. So Wiskott syndrome patients, they completely lack the protein or have reduced expression, and they get the severe immunodeficiency. And we became interested in X-linked neutropenia that was described in 2001, a large family I will show you later with 6 affected males. And it has been predicted, and biochemical data support, that they have point mutations in the GTPase binding domain that destroys this auto-inhibited folding, so that WASP would all the time be constitutively active. And these patients, the main clinical features they have is severe congenital neutropenia. But we also reason that, based on what we know from WASP in all immune cells, also other immune cells may be affected here. So just to show you some examples, quite old picture, but I think they're quite pedagogic. So this is a control lymphocyte from peripheral blood that is covered with these microvilli extensions. And on the microvilli, the cells sort adhesion molecules and also chemokine receptor that is needed for the cell to really enter into tissues. And if a cell lacks WASP, it looks very different. So this is a lymphocyte from a Viscot patient which have this very strange surface architecture. And if we look at the other side of the spectrum, again, this is a healthy donor lymphocyte covered with these microvilli. This is a lymphocyte from an XLN patient that have overactive WASP mutations, and they have a lot more dense surface architecture, and we also know that they have about twofold increase in polymerized actin. So we were interested in understanding why do XLN patients develop neutropenia. And so what we did, we started to work with neutrophils that are actually very tricky cells, probably similar to platelets that we just discussed. So you need to work with them rapidly after obtaining the blood sample. They develop in the bone marrow. It's an enormous output. They come out to the blood, and they are really the first responders when we have an infection in the tissue, for example, the skin. And they can adhere to the endothelial wall that may be activated and express some chemokine ligands. So they first have sort of loose adhesion via selectin molecules. That leads to a signal to activate the integrins that then makes them spread out on the endothelium. And then they can, because of their sort of very peculiar nucleus, that can be sort of shrink to 1 micrometer, they can rapidly go through the endothelial cell layer, get out in the tissue where they produce a lot of reactive oxygen species, they can phagocytose pathogens and kill them. So we read up a little bit on the literature, and this is the number of diseases, most of them are actually monogenetic, that affects different parts of the neutrophil life cycle. And we and here we have, for example, the lane mutations that are sort of the most common one in congenital neutropenia patients that affect early myeloid hematophases in the bone marrow. But you have also others that affect, for example, you have LAD1, that these patients lack beta-2 integrin, so they cannot make this firm adhesion, and neutrophils do not come out to the tissues. And we predicted, where our disease would be, and probably we thought it might be any of these steps, actually. So Martin Kessé, a very talented postdoc in the lab, started to work with this. And we asked 2 questions. So why do these patients develop congenital neutropenia? And how do mutations in WASP elicit different clinical phenotype? So I'm not going to show you so much, but we have actually compared these patients and mouse models with the mouse models for Viscodoli syndrome. So this was the original family with XLN that had the L272P mutation, and if you think about it, this is a leucine that induced to a proline, which really a proline will open up the sort of protein structure, the amino acid sequence. And there were 6 affected males here in this family, and we were then interested to look into a little bit more on the neutrophils in this family, and to do so, to work with neutrophils, as many as you know, we had to go to the site where the family was, and this is a family that has been taken care of by Peter Vandenberger for a really long time. So 3 postdocs in the lab basically drove down to Belgium and worked with the samples there. And what we found is that, so we basically got the samples from these 2 brothers, the sister that is asymptomatic and the carrier mother. And not so surprising, when we looked for neutrophils in the blood, this is a healthy donor, we didn't find any or very few neutrophils in these patients. And when we looked also for other markers, the Fc receptor and CD15, they basically had no defined neutrophils. We got out a few neutrophils that were there and did electron microscopy. So these are two healthy controls, and these are the excellent patients. And maybe you can appreciate that the healthy control neutrophils, they have a lot of dense granules here by EM, whereas the excellent patients' neutrophils looks quite empty, so if they already released the granules. And we could also see that they had increased the NADPH complex on their surface, which is also a sign of activation. And when we looked at blood smear, this is a control or the sister neutrophil or the mother. You see characteristic neutrophils with these nuclei. In the two patients, first of all, we had a heavy contamination of eosinophils in this prep. And then we also found cells that are actually supposed to be in the bone marrow during myeloplasia, so band cells, metamyelocytes, and other cells. And if you look at ratio here, you can see that the main contamination in these patients are eosinophils. And then we have some of these progenitor myeloid cells that is released into the blood. And then when we talked to Peter, and I should say that these patients are now in their 30s, so they are not children any longer. He said that they have a quite mild phenotype, clinical phenotype, meaning that they don't need GCSF and they don't come to the clinic that often. So we were curious to see whether we could find the neutrophils at any peripheral sites. So two postdocs went down to Belgium again last year, and we looked at neutrophils in saliva to see if we found them and if we found them in normal numbers. And as we discussed with Anna, this is not easy to do because you have such a contamination from other cells. So the epithelial cells are huge compared to the neutrophils, and they have many. So we basically we put the neutrophils through smaller and smaller columns. So in the end, we had quite a clear plot. And then we used this image stream so we could both get flow cytometry data and imaging. And the most convincing thing we found, this is now CD15, a sort of neutrophil marker, but not absolute specific. We also used a nuclear dye, TRAC5, to really try to identify cells based on a certain software aspect ratio and circularity, that they had this sort of neutrophil-like nuclei. And if we trust this, then actually the two excellent patients have fairly equal number of neutrophils to the control, the mother, and another control. So we do believe that these patients actually have neutrophils in their saliva, despite having really low neutrophil counts in their peripheral blood. Okay, but then to understand more about the disease, we went to mouse models, and so we are lucky because human and mouse wasp at the amino acid level are extremely conserved. And in the region surrounding the GTPase binding domain, it's 100% amino acid identity. It's a frame shift of two amino acids. But basically, this mutation corresponds to the human L272P, where it is shown mutations, and the I296T is corresponding to London patients with I294T mutations, and also a big Irish family, which has the I294T mutation in WASP. So we inserted them into WASP exon 9 into the germline WASP gene in mice. And I should say this nucleotide change from a C to a G took me about 5 years of postdoctoral work to do. Maybe it was my hands, but this is how it was. But now we have the mouse ones and we can study them. The other one is a CC to a TT, so very small changes in the genome. Okay, so are these mice neutropenic? No, they are not. So this was quite discouraging when we got the data in 2013 or something. So when we looked at neutrophils just by facts, Ly6G and CD11B for mouse neutrophils, they have the two mutants, XLN, have normal neutrophil numbers. what we realized is, so in Sweden we have to keep our mice very clean according to the laws. So the wild type mouse are actually also neutropenic, so we cannot really look into this, at least not in Sweden, unfortunately. But we started to, we saw some phenotypes that I will come to, so we started to look into these mouse models anyway to try to sort of really find out what was going on when wasp is in this open conformation. And yes, what we did first was actually to test, because what I told you before is a lot based on biochemical prediction. But now we had actually primary cells to see if this fitted with the sort of in vivo context. So we looked at, one prediction of it wasp in an open conformation is that it should be easily degraded if you add the protease. Another one is that the critical tyrosine here will be phosphorylated. And the third is that it will induce a lot of polymerized actin. So we took neutrophils from our mouse models and we treated them, and I should say this I actually started together with Anna's supervisor in Boston a long time ago. But we basically saw that, so this is wasp, you can see here this is a wasp knockout, so this band is missing. So this band is wasp, and when we took out neutrophils and just looked for lysosomal for wasp expression, it was completely gone. So first we thought we may have made another wasp knockout, which was not, such a nice thing. But then we realized, and this is the other mouse, the band is completely missing. When we add a very strong protease inhibitor, we actually see that the band reemerge. So we do think this shows that it's more prone to degradation, based on that neutrophils contain a lot of proteases naturally. Then we looked at phosphorylation. So here we pull down wasp, and you can see that this is wild type neutrophils. These are wasp, have no wasp, which is good. The two excellent neutrophils, they have wasp, it's slightly reduced in quantity. This is the finding we have. When we plotted for this specific phosphotyrosine, we found that in resting neutrophils, they don't have much phosphorylated wasp in the wild type setting, whereas the two excellent mutants really had hyperphosphorylated wasp. So this tyrosine was phosphorylated even without any activation. And then we look at actin. So we can look here. So here we have looked at the actin in the cortex and sort of total in neutrophils. And we have done this side by side with the wild type and the mutant. And what we could see if we look here is that both the cytoplasmic F-actin, polymerized actin, and the cortical F-actin was increased in the excellent neutrophils compared to the wild-type neutrophils, so more F-actin. So then we were interested to see what happens during these steps. Are the excellent neutrophils affected when they are supposed to activate, change cell shape and activate their integrin to adhere to the endothelial walls? So we use image stream again to look at how the neutrophils, upon activation with FMLP, for example, they will change cell shape and become polarized. And here we use a live act probe, which binds to polymerized actin. So we have bred this allele into our strains. And what we could see now over time is that the wild-type neutrophils certainly change, they become more polarized, but the XLN have a higher frequency of polarized cells. And this also correlated, we did the same thing again, just to see. So these are wild-type neutrophils, and these are the two XLN. They have a much more dense surface architecture of these protrusions, or ruffles. And then we looked at the adhesion molecules, and CD11B is the integrin molecule that is really needed for the neutrophils to adhere to the endothelial wall and spread out. And what we could find is that wild-type neutrophils have a poor CD11B expression, and then the XLN neutrophils had less CD11B expression. And this was also seen up on a pathogen infection, so E. coli, both in the blood and in the peritoneum. So they had more sort of dense architecture, they had a more dynamic cytoskeleton, but less adhesion molecules on their surface. And this was also repeated in vitro. And then we also looked at the patient cells. So here we have looked at the same integrin, CD11B. These are the dotted lines are the 2XLM patients at rest, and these are the control neutrophils. When we stimulate them, so CD11B resides intracellular in the neutrophil and is released upon activation, for example with the PMA or FMLP. So control healthy donor neutrophils will get more and more CD11B on their surface, whereas the 2XLM patient, they could not sort of upregulate CD11B on their surface. So what I told you so far is that I didn't show you the data from the wasp deficient mice, you have to trust me here. But we have shown that the excellent neutrophils have a more dynamic polymerized actin, a dense surface structure, decreased CD11B, and decreased static adhesion. I didn't show this. And we were thinking that is this all due to that they have such a densely packed cortical actin cytoskeleton so that vesicles cannot go out to the surface, for example with CD11B, and the opposite in the wasp deficient mice. So we asked a question on neutrophil functions such as migration and phagocytosis effective. And now let's see if I can show you this movie. Not sure, let's see. Not really. I just wanted to show you how rapid they move. But it doesn't matter so much. So here we have labeled actin cytoskeleton, and you have to believe me that they move really rapidly, and they actually change their cytoskeleton all the time. What we did was to use different infection models. We used Staph aureus, this is only Staph aureus, we also used E. coli, and we looked at how the neutrophils came into the infectious site in the peritoneum or in the ear. And we could see that the excellent neutrophils actually accumulated an increased number at the site of infection compared to the wild type, also here and also here. So then we wanted to test, is it so that they actually accumulate in tissues? So we used a competitive setting where we label excellent neutrophils, so the wasp knockout neutrophils, with one dye, and then not one dye, it's a congenic mark, it doesn't matter. So CD45.2, you see it here. And then the wild type neutrophils with another marker, so this would be the excellent, this would be the wild type, and these are the host neutrophils. We injected them into mice, and to really see if the neutrophil could come into the infectious site, we created an air pouch, it's called the air pouch model, which is an empty pocket under the skin with air. But that allows you to attract neutrophils. From the beginning there is nothing. If we add TNF-alpha and FMLP, that will activate the endothelium and the neutrophils will start to go out. So what we found when we looked at different organs here, so this scale basically means that if it's above 0, it's a log scale. It's an advantage for the mutated neutrophils. If it's below, it's a disadvantage. So we knew that wasp knockout neutrophils had a decreased capacity to home into tissues, both the spleen and the air pouch, whereas excellent neutrophils showed about a twofold advantage in getting out into tissues, so faster migration from the blood through the endothelium and out into the air pouch. And we wanted to see other functions of neutrophils when we looked at killing. Sorry, I'm not showing that, but they could kill as well. So the excellent neutrophils could kill pathogens as well as the healthy neutrophils. But when we looked at phagocytosis, we saw that the excellent neutrophils were always better at phagocytosing pathogens compared to the wild type. And we also measured the production of reactive oxygen species. And here we look at intracellular production of reactive oxygen species from basically within the phagosome where the neutrophil have encaptured the bacteria. And we knew from before that the wasp knockout neutrophils produce less reactive oxygen species compared to the wild type, and this is over time. The 2XLN neutrophils, they actually produce more intracellular ROS. So this suggests to us that they may sort of be in a hyperactive state, these neutrophils. Right, so then what I didn't show you migration in vitro, but we could also show you in vitro that we had increased migration and that excellent neutrophils homed quickly into tissues, increased phagocytosis, and increased intracellular ROS. So now we wanted to look into this process. We would like to mimic it using these microchannel devices. So we coated glass with P-selectin, which would be the selectin ligand, ICAM, which would provide the ligand for CD11B, and KC, which is the IL-8 for mouse neutrophils, on a glass to mimic the sort of endothelial surface. I don't think I can show you the movie. Let me try, but probably not. Yes, very good. So here you see, so the neutrophils actually do what they're supposed to do. So we put the flow pressure here on these slides. And you can see that some neutrophils are actually rolling. These are the rolling ones. And then some of them arrest, and some of them really spread out. But you can also see that they are still moving when they spread out, so they are not static. So we think the system sort of mimic the interaction with the endothelial cell layer. And then we became interested to measure, of course, comparing wild type and the XLN neutrophils in all these sort of responses. So when we increase the shear stress just by sort of forcing more liquid over the glass, we could see, so now, here we have the rolling cells. You can actually see the tracks here when you put multiple images together. And then we have a spread cell and we have the arrested cell. So when we looked at percentage of arrested cells and the increased flow, we expect them to release from the glass because of the shear stress, mimicking perhaps the blood flow. There were no difference between wild type and XLN. But when we looked at these spread cells, the XLN, so this is wild type, you see more spread cells, but the XLN neutrophils had higher number of spread cells, suggesting that they could actually adhere more firmly to sort of this glass with endothelial ligands. And finally, we wanted to see where is the active wasp located. Is it recruited to the membrane of the neutrophils? So we use image stream again. I will not go through all the details, but we looked for, again, by image stream, wasp in the membrane and total wasp. And we could find that this is a chemokine. When we activate wild-type neutrophils, more wasp is recruited to the plasma membrane. This could be inhibited with wartmanine that basically blocks the sort of lipid biogenesis of the membrane. But you can see that the excellent neutrophils already without activation had a lot of wasp in the plasma membrane. So what we think is going on here is that in the excellent neutrophils, they are hyper-responsive in tissues based on especially our mouse work or perhaps also the patient cells. And that is because WASP is all the time located at the plasma membrane where it's phosphorylated and open conformation and inducing dynamic actin polymerization. And this is needed for the sorting of all these receptors that allows neutrophil to go out into tissues. So just to show you what we are building as a little pause, this was the owl I just showed you. So now we are somewhere here, right, Nick? I think so. And this is the new research building that they are building. Across the street is the new hospital and the clinical science. And we just discussed this, but here is the bridge supposed to be that will connect the basic research with the clinical, actually the clinic patients. So we hope that will work actually. And we are supposed to be here, and we are moving. This is actually my birthday. That is our moving date. Funny enough, they actually completed the house since I took the pictures. And we were visiting there, and I just want to show this picture because I think this is how our lab works actually. So we are all in different directions, you know. We get together now in a while, and Nick is actually taking the picture here, so you're not here. There are always missing people. I think there were 2 in the clinic, one was traveling, one was working in another lab, and so forth. So this is how my lab works basically. And then here is inside. I don't know if you can see much, except for this is on the 7th floor, and I'm scared of heights. So this is me really looking down and wondering how I am supposed to work there. So it's a very nice building, but I don't know if it's made for people that are scared of heights, actually. And we are in Stockholm, so this is Stockholm, the old city. This is probably the best part of Stockholm for you that haven't been there. And if I have some time, I'm not really sure, do I? Yeah. I would like to tell you about another story, also related to the actin cytoskeleton, and this only involves patient data this time, so we don't have a mouse model yet. So what we just talked about was the WASP family of actin regulators and WASP itself. And I should also say that Nick, who is here with me, has set up tools to study WASP in the nucleus. So basically we find all these actin regulators and actin in the nucleus as well. It's quite unclear what they are doing there. And what we could find in our initial studies that WASP seem to regulate gene transcription of very specific genes. But now I want to turn into MK1, that is an actin sensor in all cells, not the least in hematopoietic cells. So MK1 binds to globular actin in the cytoplasm. It's a weak interaction. So when actin is polymerized into a filament, MK1 gets the signal to enter into the nucleus. There, it binds to the serum response factor and regulates about 100 to 150 genes, and among them is actin itself. So this is a program to really increase the actin cytoskeleton in the cell when we have low globular actin. Now, this project came from a study that was published by Adrian Thrasher's lab in 2015, where they described a girl with MK1 deficiency. So this girl had recurrent bacterial infections, quite severe, pseudomonas with septic shock, and a lot of other different skin abscesses or others. And they could, by whole genome sequencing, whole exome sequencing, sorry, they identified a mutation in MK1. And what they found in this study is that the hematopoietic cells had decreased polymerized actin, and the phagocytosis, neutrophils, was deficient. Migration is almost blocked, so the neutrophils cannot migrate, basically. And they looked at dendritic cells, adhesion, and these protosomes, these adhesive structures, and they were really deficient in dendritic cells. And then we came across, because of this study, we came across another study that was published by Magnus Björkholm at the Karolinska University Hospital in Sweden. And he described, it's a short paper, and all he described is triplets, genetically identical triplets, of which 2 developed Hodgkin lymphoma, so HL is for Hodgkin lymphoma, one in 1986 and one in 2008. They have both been successfully treated. The 3rd triplet carried the mutation, but has not developed Hodgkin's lymphoma. And they did CGH analysis and could identify an intronic deletion in MKL1. It was not in the exons for sure, but this gene, as you can see, is spanning quite a large distance of DNA. And the transcription actually starts in exon 3 or 4. So it's a complicated gene with large introns. So we became interested to see, would this intron deletion have any effect on MK1 expression and activity and B cell functionality of especially the undiagnosed triplets which hadn't developed Hodgkin lymphoma. Julian Record, who was actually the first author on the London paper, he joined my lab as a postdoc, and he worked together with Anton Sendel, and they started to investigate cells from these patients. You see it's coming, I think? No. So I just changed the color coding now a little bit, and you will understand why. So HL0 is red, and the other two are some other color, pinkish. So we looked at in primary lymphocytes from the patients, and this is based on forwarded side scatter. This is one experiment, and this is actually another experiment a couple of months later. Compared to the control, the two patients, HL0 and HL1, had increased MK1 expression. And when we did it again, it was the same thing compared to the control. They had increased MK1 expression. To really study these patients, we took the approach to make EBV transformed B cells, and we made the EBV transformation of two controls, each matched with the triplets at the same time. And Nick actually looked at mRNA expression, and we could see that the HL0 seemed to stood up. This is the non-affected triplet, and had more MK1 expression. And when we looked at protein here, HL0 is again the one with the most MK1 protein, and also here. And then we know that MK1 induces, as I mentioned, about 100 different genes when it's activated in the nucleus. So we looked at some of these, and really those that are related to the cell cytoskeleton, RAC1, TALIN, integrin, actin, and SRF itself. And this HL0 really stood out and always had increased expression of these genes, really suggesting that MK1 had more activation in the HL0 cells, the red cells. We looked at F-actin, of course, since it's an actin sensor. This is again from primary cells, the control, the two patients in one experiment. They have increased F-actin, polymerase actin, based on phalloidin staining. And this is the second experiment, same thing, the patient cell have increased phalloidin. And then we tried to set up a quite, because EBV cells and all cells have different sizes, and this is something you have to consider when you look for polymerase actin. So we wanted to look at them side by side. So here Julian set up both to look at the G-actin levels in the cell side by side and the F-actin levels. And what we could find is, if anything, that the HL0 had more G-actin, maybe more F-actin, but it didn't really reach significance. And then what we did was to see if this sort of MK1 mutation and the correlation to actin, would it lead to a more dynamic cytoskeleton? And one assay that we have used a lot since my PhD studies is to put B cells on an adhesive surface, and they will then spread out and form these sort of long protrusions here that we can measure as a functional readout for a dynamic actin cytoskeleton. So here are the control cells, and here are the, sorry, this is wrong, this should be HL0, I'm sorry. So these are the HL0 patient cells, it's quantified here. But you can see that the HL0 patient cell is actually not the patient, but HL0 cells have the most spread cells with these long protrusions, so very dynamic actin cytoskeleton, and also seen in HL1. And then we did also IRM on this, and we could find the same pattern. So this is really looking at the surface adhesion. So it seems like especially the HL0 cells had a more dynamic actin cytoskeleton. Since they developed Hodgkin lymphoma, we were interested in seeing whether we find any aberrations in cell proliferation. So we looked at the primary B cells isolated from the patients, and we looked at the sort of proliferation marker Ki67. This is freshly isolated B cells. These are the two controls, and all three patient cells had more proliferating cells based on Ki67. We looked at proliferation of the EBV cells, so starting at the same cell number, and then we found that the HL0 cells proliferated about twofold faster. And we looked at the DNA content to see if we find any evidence for increased DNA content. So a normal cell would have a G0, G1 peak, an S phase where the DNA is synthesized during cell division, and then G2M, which is when the mitosis happens. And we could see that the HL0 here had some cells, or actually 4%, 3 to 4% of the cells, had more than the 2N DNA contents, more than the normal number of chromosomes, suggesting perhaps that there is some sort of chromosomal aberration. And then one thing is Hodgkin lymphoma cells, as you may know, have this typical O-like appearance, so basically two nuclei in the Westerberg cells. So we thought that maybe this increase is due to that the Hodgkin lymphoma cells have two nuclei. So we looked at multinucleated cells, and we looked at quite a number, but we didn't find any evidence for that the controls or the HL patient cells had any difference in these multinucleated cells. They had about 1% that were multinucleated. So then we made telomere fish, which is basically a way to identify chromosomes. And you can see here, it's not the best picture, but the chromosomes are labeled in dark during metaphase, and then we have labeled the telomeres with a telomere probe. And this allows us to look for any sort of aberration, loss of chromosome content, or translocations. We didn't find anything there, but we could see that the HL0, this is the normal chromosome count in a human cell. This is the HL0 cells that had more, up to 96 chromosomes in one cell. And this is quantified here, because HL1 had a high number of this more than 2N chromosome content. And then since we work with mice, we wanted to see if we inject these cells into a mouse that basically lacks its own immune system, do the cells grow at normal rate, do they grow faster? And this is sort of a tumor model that is quite commonly used. So we injected the cells, and we looked at day 10, and we looked at the what's called tumor volume. It's not really a true tumor, but we did see that the HL0 cells, they grow much faster and actually form the tumor. And I didn't have the picture now, but the tumor actually had blood vessel formation, whereas the control cells, we could hardly find anything. And then what we did was to, let me see if this works, otherwise I think it will work. Let me see. It is so. This was just to show. Yeah, I saw that too. Exactly, let me see. It was here, so anyway, what this shows is that we started from single cells, and then we measured how quickly they would aggregate. And this is again a test both for cytoskeletal rearrangement, the growth, and many things. What we could see is that the HL0 cells, they didn't form these big aggregates. If anyone who worked with EBV cells, they form big aggregates. I'm going to show you now. Maybe the next one. But this led us to see, again, looking at a B cell integrin. Now we looked at CD11B neutrophils. Now it's CD11A, which is LFA1 ICAM interactions. So we looked at the expression of CD11A on these EBV cells. The control cells express a lot of CD11A. The HL0 cells have much less CD11A. The HL1 cell seems to have two populations, and the HL2 looked basically as the control cells. And this is just the measurement of this in MFI. The HL0 cells have really low CD11A. We looked at another integrin, ICAM1, and that was normally expressed. So it seems to be specific for CD11A. So we became curious because in a lot of the assays, perhaps you noticed, this HL0 had like a dominant phenotype, but the HL1 cells were sometimes behaving like HL0 and sometimes like the control cells. And this is the patient that was treated already in 1986 for Hodgkin lymphoma. So we were thinking maybe we could use CD11A and sort out the low and the high cells and see how they behaved. So we did that. So we sorted out the low cells, CD11A low cells, this is after sorting, and the CD11A high cells, and this is after sorting, and then we studied them. Let's see if this works. Yes, good. So these are now again the same assay. Under 2 hours we look at how the cells aggregate. This was the CD11A low cells, so they do form some aggregates, but not that much. The CD11A high cells, they form these typical EBV aggregates that is dependent on ICAM LFA1 interaction, I should say. So basically what I didn't show you before, but these behaved like the HL0 cells and these behaved like the control cells. And this is just quantified here. Then we looked at MK1 protein in the 2-cell population now from the same patients, HL1. And we could see that the CD11A low cells had higher MK1 protein compared to the CD11A high cells, and this is quantified here. And we have now injected these cells into the mice again to see if they form tumors. The CD11A low cells form large tumors. These are only 3 mice, we are repeating this right now. And the CD11A high cells form much less, sort of, if anything, tumors, so it's much smaller size. So what we think is that HL1, this patient is composed of 2 subpopulations. The CD11A low behaves like the HL0, what we believe are Hodgkin cells, and the CD11A high behave as control cells. So then we asked these questions initially, and what we could find in these patients is that at least in HL0, MK1 is highly expressed, has more polymerized actin, it induces more gene transcription, and we believe that this is the reason that these patients develop Hodgkin's lymphoma. We are really struggling to show causality. So right now we are trying to take these cells that are sort of hyperactivated and proliferating a lot, and we try to inhibit MK1 in these cells to see if we could restore the control cell function. Okay, so I raised this conundrum in the beginning. It's a big question. Many research groups are working on this. But our thinking from studying the WASP family of actin regulators is that, and as many of you know, these patients actually have a skewed immune response. So some cells are hyperactive, some cells are hyper-responsive. And I think this really shows the importance that you need to fine-tune the activity of these actin regulators in the hematopoietic cells. So with that, I'd like to thank everybody. Nick is here, didn't talk at all about his work today, about the nuclear WASP project that we are excited about. I did show you data from Martin and Julian and Anton, Ming is working with the B cell part of the XLN and the wasp knockout mice. Mariana is working a lot with dendritic cells. Joanna is working with NK cells and cytotoxic cells. You see, we try to cover all cells that we can look at. And then we have, these were 2 students that we had in the lab at that time. So thank you so much for your attention. Let me show you one last, because these are, if I can play them, this is not ours, but they are beautiful. Let me see. Yes. Have you seen these? So this is actually a real lymph node in you, sitting here right now. So this shows that the immune cells are never at rest, right? So what they have done here, this is from Gabriel Victoria's lab at MIT in the US. So they have labeled all lymphocytes with a combination of different fluorophores, and they can image them with intravital microscopy. What is really nice to show is what happens when we have a pathogen infection. This happens. So this, maybe you know what it is in a lymph node, a germinal center, so where the B cells undergo activation and, create their better antibodies to the pathogens. And what is nice to see is that there is really a selection, I cannot point now, for these sort of pinkish cells. But they are still moving, they're just moving a little bit less. So I think it's beautiful, actually, isn't it? You can imagine if something goes wrong here in the cytoskeletal responses, this wouldn't work. Very good, sorry, that was my last slide. Now.
Вопросы? Слушай, какие вопросы? Any questions? That was scary.
Sorry?
That was scary.
Yeah, that's actually.
Something crawling inside.
Exactly.
That was scary.
I think it's very good, actually.
Questions,
Yeah.
I don't know if you can answer.
Yeah.
So, if I understood correctly, all 3 siblings, triplets, they have the same mutation.
Yes.
So what protects the HL0?
No.
It seems to be the inverse.
Exactly.
In vitro studies, and yet it doesn't happen.
Exactly. And we are thinking that, why he didn't develop it, we don't know. But we do think we see the sort of pre-lymphoma cells in him. Whereas the other 2, the HL2, where we don't see this phenotype, was treated in 2008. So we're thinking that, maybe the cells are coming back. I agree, actually. But he didn't develop. Yeah.
I think they might have another mutation that would actually distinguish them
Yeah.
with HL1 and the other 2.
So we discussed this a lot, and first I should say that this is a heterozygous mutation. So it's all of one allele, which complicates things.
Yeah, but what I'm saying, it might be that there is another gene
Yeah.
modifier that would make them different, that is HL1 and 2 without mutation, HL01, and even though they have the same,
Yeah.
but they have something.
I agree with you. I think that so.
Maybe having mutation
Yeah.
is not enough.
No, that could be. So they are. So the clinician tells us that they are genetically identical. So it may be a somatic mutation in that case they have acquired. And we have not performed whole genome sequencing. We try to identify the breakpoint. Now we're just talking about MK1 to really identify the lesion. And Nick worked quite hard on that, actually. And from that sort of, because we run a lot of PCRs on this locus, we could see that the triplets have a unique sort of bands. It's not to say that they're completely genetically identical, so we don't know. It could be. We haven't done whole exome sequencing, for example. The thing is, this is also an intronic mutation, so it's a bit difficult, actually. But it's a really good question, of course. This is what the reviewers ask. So, yeah, maybe it was you, Anna.
Yes, where. So another question also going down about neutropenia. So you seem to imply that those patients are not truly
No, they have.
somewhere,
Exactly.
not just not in blood.
Yes.
But the controversy in that theory is that the bone marrow is not
Yes.
So
Exactly.
basically, where do these neutrophils come from? I think they're somewhere. They don't come from bone marrow. Where do they come from?
Yeah, and we could actually show, we have, I didn't include it now, but we have done, so we purify CD34 cells from the excellent patients and the controls. I actually don't think I have the data, but so first of all, they seem to have more CD34 phosphohemolytic stem cells in their blood. That is also strange, I think. But then they couldn't give rise to neutrophils. So we still don't understand why they are in tissues if they are in tissues. So it's a combined, the saliva maybe is difficult to see, but it's more the clinical symptoms.
Patient was not treated with disease.
No, they are not actually, and neither is the London patient. So the I2940 don't get G-step as well. And there's also this large pedigree from Ireland, which has I2940, and they have a very variable clinical phenotype. It's a lot of affected boys or men in that family, and despite having these really low neutrophil counts in blood, they have a very variable clinical sort of spectrum. So maybe modifying genes also, at least in the families. Could be. And we were also thinking that if it's so, which we are quite convinced that the neutrophils spread out much better on sort of endothelial ligand, it could be that we're measuring a blood sample, but the neutrophil is actually present on the vessel wall. Exactly. Could be. So, and the third that we haven't investigated at all, but that we sort of would like to go into is extramedullary hematopoiesis. So it occurs in, better than me, certain sort of clinical situations, you have this extramedullary hematopoiesis that can really produce neutrophils. So maybe.
There is no clear evidence for that in this patient.
No. Yeah, exactly. Yes. Can take it in Russia, maybe I understand. Don't know. That's a while ago. But yes, thank you so much.